All right. Well, welcome everyone. Thanks for joining us for our November Brain Stimulation Journal Club. I'm Ellen Suter. I'm a PhD student at the University of Minnesota. I'll be moderating this session. I'm also a physical therapist, and so I'm really interested in how neuromodulation can be most effectively used in clinical applications for motor learning and rehabilitation. So I'm excited to hear from our speaker today on the topics of advances in neuromodulation for motor plasticity. Just a few housekeeping things. We are recording this session. Keep your microphones muted during the talks. You're welcome to type questions into the chat at any time, and I can relay them to the speakers. Or after the talks, we'll have some time for question and answer, and you're welcome to unmute yourself then and directly ask your questions too. Prior to starting with our speakers, I'd like to welcome Dr. Mark George and Dr. Marone Bixon, who are editors of the Brain Stimulation Journal, to present a few opening remarks and announcements about the upcoming International Brain Stimulation Conference.
So hello everyone. And Mark, please let me ask you to maybe give people an overview of what you're expecting for the meeting. If there are any questions, we can address them. I know the meeting is full and going ahead as planned, but I don't know if you wanted to sort of give people more of a what to expect. Looking forward to the next couple of weeks.
Yeah, thanks. As everybody knows, there's a pandemic and it's made everything 5 times more complicated. And so we had originally scheduled the 4th International Brain Stimulation meeting to go happen in Kyoto, Japan, back in February. And there have been 3 before, each better than the one before. The last one was in Vancouver with about 1,000 participants and 60 vendors or industry representatives, and just a wonderful kind of eclectic bringing together of the brain stimulation community from all over the planet. And we rotate the meeting around Asia, Europe, and then North America. And so this was supposed to be the Asia meeting. It got rescheduled to December in the same venue. And then last summer, it became very clear that Japan was not opening up in the way that people had thought. And so Elsevier Meetings, who sponsor it, said, we'll just make it all virtual. And Harold Sackheim, the co-scientific chair, and I argued that, no, we think America is worth taking a bet on North America. We should move it here. And oh, by the way, Charleston might be a nice place. And so it was moved to here in Charleston, and it's going to happen. We had about 1,000 posters and people and oral presentations. And we actually so far have around 600 people who've registered, and then there are about 35 industry groups, and I'm not sure how many within that. And so it's filled up wonderfully, and the science should be as good, if not better than before. As you know, the journal is aggressively agnostic with respect to which of the brain stimulation devices to study. The premise is that they all have commonalities, and that if we understand how stimulation interacts with brain, regardless of whether it's TMS, TDCS, VNS, that's the science that we want to push towards. And so it involves presentations across all of those. So it's going to happen. We have at least 600 people who will be here. The hotel is full. We got a smaller meeting space than normal because we didn't think we'd hit 1,000. And so, but it's safe. So we're going to, have full COVID protocol. You have to be vaccinated and wear a mask, and there's spacing. But it's going to happen. So I encourage you to come to Charleston if you haven't registered already. And rates of COVID in South Carolina are as low as they've ever been. We've gone through the Delta surge, and it's pretty low. It is around, so we have to be safe. But I encourage you all to think about it and come and have fun. So thanks for letting me reassure everyone that there's going to be a great meeting. as you know, with Marome and some other extremely talented deputy editors, we published this journal, and it's great, and the science has just rolled along, but it's not quite the same as actually an in-person meeting. And I'm for one have missed kind of the poster sessions and the rubbing of elbows and the things you learn at an in-person meeting. And so I'm looking forward to it.
And I just want to add, I think these in-person meetings are especially important for people who maybe are earlier on in their career, the kind of people we want to also showcase here in the Journal Club. So that networking, you know, is even more important. And so I think that's one of the impetuses for making sure that in-person meetings don't completely stop, right, for 5 years, and we keep having them. And I certainly know that when I was starting my career, one of the most important moments was when some guy I did not know with a mustache came up to me. I had some lonely poster at the end of a corridor, and he came up to me and told me about a journal that I never heard of. It was a new journal at that time called Brain Stimulation, and he suggested I submit my poster as an article to it. So these meetings, these in-person meetings, can have little small moments that are career changing. So that's why.
Yeah, I remember the moment as well. It was in Göttingen, Germany, at one of the brain stimulation meetings that preceded these international meetings. Kind of these are in the tradition of that, and that began a whole series of publications about modeling within the brain and brain stimulation. And as some of you may know, just like Alfred Hitchcock would always kind of bury himself in a little scene in every one of his movies, somebody on this Zoom right now with the initials MB has had his profile on the cover of Brain Stimulation. I've lost count. I think it's 6 now.
Is
that right? I get to choose the cover art. So whenever I can, I try to add to that number.
Great. So I don't know if there are any questions about the conference. We can certainly address them now. You can certainly email me, I think, and Dr. George also directly, but certainly feel free to email me. And if I can't address the question about a conference, I will get you the answer. If you have any concerns, you can always email us as well. I don't know if there are any comments or thoughts for the meeting. So if not, I guess we can go back to the Journal Club moderator and have her take it away.
All right, sounds good. Thanks so much, you both. Okay, so we'll jump into the talks now. We have 2 speakers today who will both share their recent work with you. We'll have a couple minutes of questions after each talk, and then we can take more questions about both talks at the end if there's time. And again, you're welcome to type questions into the chat at any time, and I can read them out to the speakers at the end of the talk, or you may also unmute yourself after the talks to ask your questions. Okay, so I'm excited to introduce our first speaker, Dr. Sarah Hussein from the University of Texas at Austin. And Dr. Hussein will be presenting her recent work published in Brain Stimulation titled Phase-Dependent Offline Enhancement of Human Motor Memory.
All right, so I'm gonna share my screen. Make this big. Okay. Slideshow. Okay, y'all can see that?
Looks good.
Okay, I'm starting to say y'all now that I'm in Texas, so yeah. It's just a thing. Okay, so thank you, Ellen, for introducing me. Thank you guys for inviting me to talk about my work at this journal club. So I'm going to talk today about a few recent projects. So I will discuss the paper that was the basis for this invitation, but I also want to talk a little bit about applying brain state dependent stimulation in the lesion brain and some methods that might help us with that. Most of this work was carried out at during my postdoc in Leo Cohen's lab at NIH. But the very last bit, which is like super short presentation, is work that I've done in my own lab here at UT Austin, hence the burnt orange theme of my PowerPoint presentation. So first of all, there we go. First of all, a brief outline. So I will first discuss the paper that was the basis of my invitation to speak here. The topic on this is how phase-dependent TMS affects human motor memory in the healthy brain. And then afterwards, I'm going to shift gears a bit to talk about how we might develop personalized state-dependent TMS for the lesion brain. My particular interest is post-stroke. So let's start with the paper. So of course, humans learn a variety of skills every day. Many of those skills, particularly procedural skills that involve linking together well-learned movements into new patterns, show offline changes. And what I mean by offline changes is that performance will improve from the end of one training session to the beginning of the next training session, sort of away from actual task practice. That may or may not involve sleep, but the idea is that skill will improve in the absence of practice. This is what we call offline skill learning, and it engages the primary motor cortex, even during waking rest when an individual is awake. However, primary motor cortex activity is not consistent or stable or constant over time. Actually, it oscillates in the mu range, and this is between about 8 to 12 hertz. And one of my favorite studies that shows this sort of oscillation and how it affects the motor cortex was done by Saskia Hagens. So she recorded local field potentials and single neuron spiking rates from the primary motor cortex and other motor areas in non-human primates. And she showed that at the peak phase of an 8 to 12 hertz range oscillation, spiking rates were at their lowest, whereas at the trough phase or the bottom part, spiking rates were at their highest. And since this work, there's been a wealth of work showing that these different mu phases also co-vary with corticospinal excitability as we measure it using single pulse TMS. So I think the interesting thing here is that these peak and trough phases are really kind of physiologically distinct states. And if they are indeed physiologically distinct, then the way that they affect or contribute to motor skill learning, particularly offline learning, might vary across these phases. And along these lines, there's also a wealth of work in the perceptual and cognitive domains showing that visual perception, tactile perception, attention, those all oscillate synchronously with local brain rhythms such that those functions are maximized during specific brain rhythm phases. So for me, this brought up the question is, does motor skill learning, and particularly offline skill learning, follow a similar phase-dependent principle? Does it vary according to the phase of this mu rhythm oscillation? So we aim to address this, of course, with an experiment, as we do. We had healthy participants come to the lab for 2 consecutive days. On day 1, they all came to the lab. We set them up with EEG, EMG, TMS, and then we had them perform a motor sequence learning task, which I'll talk about in a second. After they completed that task, we delivered a phase-dependent TMS intervention that was about delivered at 1 hertz, and TMS was either delivered during mu peak phases, shown in red here, or mu trough phases, shown in blue. And then in a third group, we delivered sham or placebo TMS that didn't actually activate the brain, but delivered sort of that tactile sensation of a tap and the auditory sound during random phases. And the reason we did this during the period after sequence learning is we think this is when offline processing that contributes to offline learning is occurring. So we wanted to sort of affect this process in different ways, depending on whether TMS was delivered during peak, trough, or this placebo was delivered during control phases. Then the next day, they came back to the lab and they reperformed that sequence task for us. So the sequence task that we had them do was an explicit motor sequence learning task. It's very common in motor sequence learning literature. And they had to do 36 trials of this task. Each of those trials alternated between periods of typing and periods of rest. So they saw a short ready screen followed by a go cue. Then they were presented with a string of numbers that was the same string on every single trial. And their job was to repeatedly type that sequence of numbers as many times as they could, as quickly and as accurately as possible in that 10-second period. Then afterwards, they got a rest for 10 seconds and the whole trial would repeat, and that would be the next trial. So the first thing we needed to do was, because this is what is sometimes called closed loop or brain state dependent TMS intervention, we needed to make sure that our algorithm could accurately target mu peak, mu trough, and random phases in our group of participants. So the way we do this is we allow the real-time algorithm to identify the target phases and sort of send a trigger to the EEG system, but we don't actually deliver stimulation. And so here I have in black the time at which the algorithm identified each phase for each group. So the blue is indicated for trough groups. This is the grand average across all trials and all participants in the trough group. The red is the peak group, again, grand average, and then the green is the control or that random group. And so you can see that we're targeting pretty well the peak phases for the peak group, the trough phases for the trough group, and the random phases for the random group. And the random phases ends up leading to sort of an averaging out of any oscillation, which is why the line looks pretty flat. On the top here, it's just the raw EEG data. And then on the bottom is the data filtered into the mu or 8 to 12 hertz range. And then also I just have some phase angle histograms, again, showing that targeting of peak phases in the peak group was accurate around 90 degrees, which is how we defined peak phases in our data. And then for the trough group, that was accurate around 270 degrees. And then the control or the random targeting was evenly distributed around the unit circle. So this tells us that our real-time EEG targeting is working properly, and any conclusions we make, we can consider that in those conclusions. Okay, so let's look at the behavioral data. So this is acquisition of the skill on day 1. So 1 through 36, that's the trials. Each group is shown in a different color trace here, and you can see the key. And we can see our dependent variable is the number of correct sequences typed per 10-second training block. And all 3 groups seem to acquire the skill pretty similarly on day 1. After they acquire the skill, we give them the phase-dependent TMS intervention. This is the actual data from the phase-dependent TMS intervention. So this is the grand average of peak targeting during TMS, grand average of trough targeting during TMS, and grand average of random targeting during placebo TMS. Then the next day they come back, but we actually start to see a little bit of divergence in their performance here, and we're going to unpack that going forward. So we first confirmed that the acquisition between the beginning of day 1 and the end of day 1 was similar across all 3 groups. And then we looked at offline changes. So remember, we're interested in what happens from between here and between here. And when we do that, we compare late day 1 to all of day 2, and we see that all 3 groups do show an improvement over this offline period. And but that improvement varied between the 3 groups. So you can see the green line for the control group and the red line for the peak group are kind of parallel to each other. They're changing similarly, but the blue line for the trough group is increasing at a faster rate. And so we saw that delivering phase-dependent TMS during trough phases improved offline learning compared to control random stimulation, which is our placebo, and then compared to the peak phase as well. So trough was different than peak, trough was different than control and peak were not significantly different from each other. So from this, we can conclude that delivering phase-dependent TMS during mu rhythm trough phases, but not peak phases, boosts offline skill learning. There are a few potential mechanisms for why this might be. These are the 3 that I think of. It doesn't mean that they're the only 3. I'm sure there are ones that I have not considered. The first is that delivering TMS during these mu trough phases increases induction of M1 LTP like plasticity, and that increased plasticity drives greater offline learning. A second possibility is that mu trough phases reflect some pulsed inhibitory state that normally inhibits offline learning, but then when you deliver TMS during that inhibitory state, you disrupt it, allowing offline learning to proceed uninhibited. And then a third possibility is that these mu trough phases are actually excitatory, and by delivering TMS during them, we're sort of maximizing that excitatory process, which then drives greater offline changes. Now, from our data, we really can't distinguish between these 3 mechanisms, but regardless of the mechanism at work here, we can conclude that these mu troughs represent windows of opportunity during which TMS can preferentially strengthen motor memories, which presents some exciting opportunities for potentially rehabilitation applications. But for me, I'm interested in the post-stroke brain and rehabilitation and motor recovery. And so one of the things that I was thinking about as I was doing this work is, well, like, how will this work in a post-stroke individual? And so that led me to ask the question, well, what about the post-stroke brain? Can we even do this complex real-time phase-dependent TMS technique in a brain that may have lesions either cortically or subcortically? So this leads me into sort of the second portion of the talk, where I want to talk about developing personalized state-dependent TMS for the lesion brain. So while I was completing the work that I just presented to you, I also was carrying out a really, really small study. It's only 3 people that was published as a letter to the editor in Brain Stimulation. And we evaluated whether phase-dependent TMS could be delivered accurately to the lesioned hemisphere after stroke in chronic stroke patients. And we found that it could. We could do so just pretty much almost as well as we did in healthy individuals. But despite those exciting, like, technically feasibility results, one patient sort of stuck with me that we tested. So this is patient A from that paper. And she had a very large, as you can see, lesion that involved subcortical structures as well as some cortical structures. And so when we recorded her EEG, we saw a power spectra that looks kind of mu-ish. So the mu is delineated by 8 and 12 hertz here. This is her power spectra over that lesioned hemisphere. And it looks mu-ish, but it is a little bit shifted, a little bit lower than you might expect if it was a pure mu rhythm. Regardless of this slightly slower mu rhythm, we could accurately target it with the phase-dependent algorithm. So again, this black line indicates the time of phase targeting. And for this patient, we could accurately hit her peak phases and her trough phases, and then her random phases were a little bit more distributed. So on the surface, technically, this seems to be working reasonably well. But if we're thinking about it, she has this huge lesion, right? And so we don't really know what we're recording, if what we're recording is actually a mu rhythm. First of all, it's a little bit slower. Second of all, where exactly is it coming from? She has this lesion. It's likely filled with lots of CSF that can alter the normal conductivity from the brain to the scalp. And so that's sort of puzzling. It's hard to know what exactly we would be stimulating if we delivered stimulation during this phase. So for me, this raises a problem. It shows that phase-dependent TMS in the lesion brain is technically doable. Like, we can actually do it in the lab, we can make it work, but it's physiologically challenging in the sense that we don't really know if the rhythm that we're recording is actually the mu rhythm. And even if it is actually the mu rhythm, that mu rhythm may not relate to spiking or corticospinal excitability in the same way that it does in a non-lesion brain. And so one possible solution for this is to develop data-driven personalized methods that discover brain states associated with high and low M1 excitability. So this last bit is the part that I've done more recently. I don't have too much time to show you too much, but I just want to show you a little bit. So we, to try to address this issue, trained machine learning classifiers to discriminate between EEG brain states during which TMS evoked either large or small motor evoked potentials or muscle twitches produced by single pulse TMS. And we did this in healthy individuals. We do have some stroke work ongoing, but I'm just going to talk about the healthy people for now. And when we did this, we found that classifier performance, so the ability for these trained machine learning classifiers to successfully discriminate between high excitability and low excitability states, performed above chance, which is shown here in this gray dotted line, in every single subject that we looked at, which for me was pretty exciting because maybe some people have effects that work in every single person, but usually that doesn't happen, at least in my experience. So we found that this seemed to work in everybody. And then when we also looked just as a gut check to make sure that the MEPs elicited during classifier predicted high excitability states, whether those were larger than those elicited during classifier predicted low excitability states, we again found that this was the case, meaning our classifiers are working properly in every single subject tested. And the average MEP modulation between those predicted states was about 25%, which is actually quite consistent with what we see with mu phase dependent TMS. And finally, I don't have the data to show you here, but we also tested how these classifiers worked on individual subjects versus the entire group. And we found that these classifiers really performed best when using subject-specific information or subject-specific EEG activity to identify high and low excitability states. So overall, even though I haven't been able to go too much in detail with this, that work shows that individual healthy subjects exhibit unique scalp activity patterns that predict low and high M1 excitability states. Now, yes, this is all in healthy people, but there are some specific advantages to this approach that might make it particularly well suited for the lesion brain. So because it's purely data-driven, it doesn't require that the brain has any specific structure. You kind of just work with what you've got in terms of the EEG signals. So far, it works in every healthy subject that we've tested, and we've tested it in 20, which is exciting for me. And then finally, we're looking at this in the lesion brain as well right now. And it seems to be working actually even better. The classifier performance is much higher in the post-stroke brain than in the healthy brain, which was super surprising to me. I didn't expect that. Those, of course, are very preliminary results, and we'll sort of have to see how those play out. But I think it raises the possibility that this personalized approach can be particularly useful for the lesion brain when trying to develop interventions that might improve post-stroke motor behavior and could interact with rehabilitation approaches. So that's all I have really for today. I just want to acknowledge my postdoc lab, which was Leo Cohen's lab. That's at the Intramural Research Program of NINDS. They were my funders. We also collaborated with the Zeeman lab at the University of Tübingen. And then some of my students are working with me on the post-stroke data right now in my lab at UT Austin. And of course, I have the references for the papers I've talked about here. And then I also just like this figure because except for maybe the do this should be at the trough instead of the peak. So yeah, that's all I have. I'm happy to take any questions.
I would figure just jump in with, thank you for that talk. This is really nice work.
Thank you.
I wanted to ask you, so separate from sort of like the scientific elegance of sort of figuring out when you should stimulate, if you went back to the dumb approach and just stimulated, I don't know, 10 times, is there a cost to doing it the dumb way? Like if someone were to say, well, I'm not going to do closed loop, but I'm just going to stimulate 100 times more.
Yeah. Yeah, I think that there is. I don't think there's been any studies showing this, but so some of the work from Ulf Zeeman's group where they show that delivering TMS during troughs produces LTP-like changes, where delivering TMS during peaks induces some weak LTD-like changes. So if you imagine you're doing like an open loop random TMS intervention, you're basically hitting a mix of peaks and troughs. And so any LTP that you might induce by hitting a trough, you're then kind of like making it go the other way with potential LTD by hitting it during a peak in a second pulse, right? And so you're kind of, it's like you're shooting yourself in the foot. Like, yes, you might hit the troughs and that's good, but if you hit just as many peaks, you might undo most of what you already did. But again, I mean, that hasn't been specifically studied. That's kind of just my read on the situation.
So, but in your case, there's an optimal timing,
yes,
which helps, but the non-optimal in your case doesn't necessarily necessarily cost you anything?
Right. So yeah, I guess in the motor learning case, it doesn't seem to cost anything. That's true. Yeah, that's a good point. I hadn't thought about it that way.
I have a question too, and everyone feel free to type any other questions into the chat or also unmute yourselves. But I was curious, so you chose to apply the brain stimulation after doing the motor learning task. I'm wondering, I'm assuming that was kind of to target that memory consolidation phase. Do you expect that you would see similar results if you had done it before or during the task instead of after?
So yes, I'm not sure. Yes, that's a great question is what I mean to say. So I'm not sure. I think it gets a little bit at the idea that, so some of Leo and Marlene Bonstrup's work has shown that consolidation might be happening in these little gaps between typing and rest. And so if that's true, then delivering stimulation beforehand might enhance that process. But I think it depends on the mechanism. So, right, I presented 3 mechanisms. One is like the increased LTP, like M1 plasticity effects that then drive those changes. If that's the mechanism, then doing the stimulation beforehand, I would think would work, right? Because you're sort of elevating M1's function, which then produces stronger offline, potentially online learning as well. But if it's a interaction with a post consolidation process, then that might not work because you need to put the brain into that consolidation state in the first place for the TMS to successfully interact with it, right? Which is a really good idea for how to get at which mechanism might be the case. Yeah.
Can I have a question? So you did your test phase on the day after the initial practice session, right?
Yeah.
Did you try doing it kind of after a gap, so to speak, so you could take apart the kind of idea that some of this is going to happen during sleep and some of this is going to happen during non-sleep, so to speak?
Right. So that, honestly, if we were redesigning it now, we would totally do that. But hindsight is 2020, right? But having like an immediate retention, sort of immediate waking consolidation, and then a sleep-based consolidation test would be great, because then we could attribute this to, is this a waking? Even though we're giving stimulation during waking, we don't really know if it's only affecting the waking process, because there is an intervening night of sleep there. So we didn't do that, but I agree that it would have been an excellent design choice.
That's the next experiment, right?
Yeah.
All right, just for timing, we'll move on to our next speaker now, but thanks very much, Sarah. That was a great talk. And if anyone has more questions, we can address them all at the end as well. Okay, our next speaker is Matt Waitman from the University of Birmingham. And his recent work published in Brain Stimulation is targeted TDCS selectively improves motor adaptation with the proximal and distal upper limb. Go ahead, Matt.
Thank you. Is that okay? Everyone see that?
Looks good.
Perfect. So yeah, thank you, Ellen, and thank you very much for the invitation, and also Sarah for that really interesting talk as well. So similarly, I'll talk about the paper that we published and a little bit of follow-on work, but not quite as extensive as Sarah did there. So a tiny introduction to me. So I'm a final year PhD student at the University of Birmingham here in the UK. And my focus of my PhD has really been kind of combining sensory motor adaptation with non-invasive brain stimulation. So looking at ways we can use non-invasive brain stimulation to modulate performance. And what we've really tried to focus on using our knowledge of kind of the central nervous system structure and function, and then also foundational mechanisms of kind of motor learning and motor adaptation to improve stimulation protocols. Which kind of brings me to kind of the focus of the presentation today, is where we try to improve the specificity of stimulation by specifically targeting brain areas or kind of brain circuits, which may have preferential control over different behaviors performed during these motor adaptation tasks. So as I'm sure many of you know, transcranial direct current stimulation is a non-invasive brain stimulation technique that passes weak electrical currents through the scalp from electrodes and via kind of sub-threshold alterations in neural membrane potentials, modulates excitability and spontaneous brain activity. And due to kind of this capacity and its capacity to induce plasticity in different areas of the brain, it's kind of been a popular research tool with kind of the hope for clinical translation. And in a research setting, it has shown to improve various aspects of upper limb function and learning in kind of healthy young adults, older adults, and then also patient populations. However, more recently, there seems to be kind of a growing number of studies reporting mixed or null or inconsistent results. And the reasons kind of as to this are relatively unknown, but it might be due to the fact that understanding how these changes in excitability translate to modulation in complex behaviors limits our understanding of how we can best apply the stimulation. And also, it's not exactly clear that all the behavioral tasks used in these studies are chosen to kind of highlight the contribution of the brain areas that we target with stimulation. And these kind of conflicting findings are notably evident in the field of motor adaptation. So we have kind of seminal work from Joe Gallagher and colleagues, and then also Hardwick and Selnick with older adults who have shown that if we apply stimulation to the anodal stimulation to the cerebellum, we can improve motor adaptation, shown here in this kind of red line. And then this is also the same for older adult participants here in this black line. So they were improving motor adaptation, and it was proposed that stimulating the cerebellum enhance these kind of spatial realignment or forward model recalibration processes. However, kind of slightly more recently, in a different lab in Oxford, using a really similar study design, they showed that actually when applying stimulation to the cerebellum, there was no effect on adaptation, but instead, when they applied it over the motor cortex, they improved adaptation in both younger and older adults, shown here in this blue line. So they kind of a really contrasting result depending on the different studies. And there were kind of a number of differences between the study designs, but what the authors highlighted is that the potential conflict could be due to the differing effectors used during the tasks. So the former used a joystick, which was manipulated using dexterous hand and finger movements, whereas the latter two used either a digitized pen or a robotic manipulandum, which required whole arm multi-joint movements to complete the task. And if we think about kind of the control of the upper limb, we know it's really important and vital to carrying out even the most basic activities of daily living. And normally we smoothly integrate the control of the hand and the arm. But there is kind of some research to suggest that the proximal and distal portions of the upper limb may be subserved by somewhat distinct neural substrates. And this was potentially most notably highlighted by Dons Lawrence and Hans Kuipers in the 60s. And they did some animal research where they severed the cortical spinal tract in monkeys at the level of the pyramids. So this left the motor cortex intact, but it removed its direct influence on the spinal cord. So after the surgery, obviously motorically, the animals were kind of severely impaired, but after a relatively short period of time, they were able to kind of recover the ability to walk, run, and importantly, use their arms to reach through kind of the gaps in their cages. However, what they never kind of fully recovered was the ability to independently move their hand and fingers, and they always had to use their whole hand in concert. And then in contrast, when they lesioned kind of more medially descending pathways originating from the cerebellum and the rubospinal tract, they found that these proximal limb movements, so those reaching movements, were the ones that were more severely impaired. So from this, we kind of can come up with a kind of reductionist framework where we say kind of the M1 and corticospinal tract seems to have this preferential control over fine hand and finger movements, whereas kind of the pathway of the cerebellum and brainstem seems to be more interested in controlling these arm movements. And we kind of see this dissociated recovery of proximal and distal movements, which has just been displayed kind of post-stroke in humans, and then also in some early research when they used experimental kind of surgery on the motor cortex to treat epilepsy as well. So with kind of all of this background in mind, both kind of that neurophysiological and the stimulation adaptation, we wanted to see if we could kind of clear up some of this confusion. And we wanted to combine targeted stimulation of either the cerebellum or the motor cortex and target those with tasks that share similar motor control characteristics, but isolate whole arm movements from these kind of dexterous hand and finger movements. So we recruited 90 younger adults and 76 older adults. And we first initially split them into tasks which either promote those dexterous hand and finger movements using this handheld joystick, or these multi-joint reaching movements using a robotic manipulandum. And they were then subdivided into either an M1 stimulation group, a cerebellar stimulation group, or a sham group. And what we got participants to do was complete a pretty standard visuomotor adaptation task. So they had to use the apparatus of the various tasks to direct a cursor towards visual targets, kind of shown here. And the aim of the task was just to direct the cursor as accurately as possible. We then introduced a visual rotation of that cursor, which results in the movements becoming accurate. And this drives the motor adaptation process, which means movements become slowly adapted and more accurate again over the course of the task. So we had baseline phases at the beginning of the trial, where participants received veridical feedback. We then introduced the rotation in the first adaptation phase, where participants then experienced this visual rotation of the cursor. We followed this by a break period to see if we could get any kind of follow-on consolidation effects, where they then immediately afterwards re-entered this adapted state, followed by some washout trials. And during the experiment, participants who were in those active groups received 17 minutes of anodal stimulation during the baseline and adaptation phases, and those in the sham group had a really short period of stimulation, which was our sham protocol. And given kind of the proposal of these differing levels of control exerted by M1 and cerebellum over proximal and distal muscles, we hypothesized that those who received cerebellar stimulation but not M1 stimulation would see improved performance when they were using these reaching movements, and conversely, those who received M1 stimulation would not cerebellar would see an improvement when using these fractionated hand and finger movements. And then also, as there's evidence to suggest that these regions undergo age-related change, we wanted to examine if this double dissociation kind of held in the group of older adults as well. So to first look at the younger adults, here we've just got kind of the time course of the study. So we've got the rotational error, so how far away they were from the target on the y-axis, and then the x-axis has just got the trial number. And what we can see at the very beginning is when participants are in these baseline phases in both of the tasks, they're able to really accurately direct the cursor towards the target. Then when we introduce this 60-degree rotation, we can see participants in all of the groups make kind of large errors in movement. But then gradually over the course of the adaptation phases, they're able to adjust their movements and become more and more accurate again. What we can see really obviously in this first adaptation phase is when participants were doing this VBOT task, which involved those reaching movements, when we applied cerebellar stimulation, we can see an enhancement of adaptation. So they were able to reduce their error more so, especially during that first adaptation phase, which was almost the opposite when they were using these different participants were using these hand and finger movements. We saw an improvement in performance following M1 stimulation. The effects after the break were less so, and we kind of suggest that there's some kind of offline learning and then also some ceiling effects due to the fact these are kind of healthy young adults. And then just focusing back in on that first adaptation phase, what we did is we calculated the area under the learning curve, which gives us kind of a measure of total error. And we can see in this kind of task that involves reaching movements, the cerebellar stimulation resulted in significantly less error during the task compared to the M1 and the sham group. And then conversely, for that dexterous hand and finger task, we can see M1 stimulation boosted performance compared to the other two groups. And then investigating whether this effect held true for older adults. And kind of the simple answer is really nicely, yes, we see this improvement in the reaching task following cerebellar stimulation, particularly towards the end of the first adaptation phase. And then that did follow on into the second one, which hopefully kind of suggests that ceiling effect might be present in the younger adults. And then less so in the joystick task, but we did see M1 stimulation improving performance in that task that involves those hand and finger movements. And again, when we look at the area under the curve, it's a really similar picture, showing this double dissociation between the stimulation site and the kind of effector used to complete the task. And visualizing that slightly differently, I think it kind of shows really clearly that double dissociation, especially here in the younger adults, where we see the cerebellar versus M1 in terms of the effectors that they use during the task. The older adults seem to perform better. They found the task using the joystick much easier, so they performed less error during those tasks. But when we combine it for kind of age group, we can see really nice hold true of that double dissociation, which proves significant as well. So moving on from that, that was the study that eventually got published. We then wanted to kind of confirm these results, and in the kind of the most simplistic way we could, we thought if we reverse the polarity of stimulation, so instead of having polarizing anodal surface positive, we wanted to use cathodal surface negative stimulation. And in kind of simple terms, when we got participants, a new set of participants to complete that task that involved reaching movements, when we applied cathodal stimulation to the cerebellum, we can see really nicely that we have this impaired adaptation compared to that darker shaded black line, and then also that kind of stimulated control M1 group. So that relationship between stimulation and effector held true when we reversed the stimulation polarity. When it comes to the tasks that involve those hand and finger movements, it's a slightly more confusing story. Anecdotally, we can see in this red line that we see kind of impaired performance from cathodal M1 stimulation, which is as our hypothesis, but this was kind of unreliably significant and mostly was not different from the sham group. But what we did see, and quite surprisingly, that when cathodal stimulation was applied to the cerebellum, we saw this kind of vast enhancement of adaptation performance compared to the sham and the M1 group, which is at first relatively surprising, but we took some time to think about it. And we know it's well established that there are functional connections between the cerebellum and M1 in the form of the cerebello-thalamocortical pathway. And this is one of the major outputs of the cerebellum. And at rest, this pathway exerts an overall inhibitory tone over the motor cortex, which is termed cerebellar brain inhibition, or CBI. Research has shown that TDCS can modulate CBI via modulating Purkinje cell excitability. So anodal stimulation is thought to increase cerebellar brain inhibition, and cathodal to decrease it. And this can be measured by kind of a paired pulse TMS design here, so we can see whether the MEP from the motor cortex is increased or decreased. And with this knowledge, we speculated that if we're applying cathodal stimulation to the cerebellum, we are decreasing cerebellar brain inhibition and therefore increasing the excitability of M1, and thus kind of indirectly facilitating adaptation of those kind of dexterous hand movements in a similar way to when we just purely added anodal stimulation to M1. So it's a kind of a direct and an indirect effect of the different protocols. And then this is just kind of highlighted here in this framework, where we see the results from the anodal study, this double dissociation depending on the stimulation site and the effector used to complete the task. And then when we reverse the polarity of stimulation, we saw this kind of indirect effect, and then also a direct effect of cathodal stimulation, and some anecdotal evidence in terms of M1 stimulation, but didn't prove significant in the end. And from these kind of series of experiments, what we conclude is that stimulation of different neural substrates, which may exert somewhat divergent influence over upper limb movements, can drive better adaptation in the tasks which share similar motor control characteristics, but isolates those kind of whole arm reaching movements from those really fractionated hand and finger movements. And it's always quite difficult to speculate the exact mechanism in play during these kind of complex behavioral tasks, and especially as it is also unknown how TDCS modulates these complex behaviors. But it seems to kind of align with some of the theoretical proposed effects of TDCS, and specifically that the kind of activity selectivity mechanism, which suggests that stimulation is preferentially modulating circuits in the brain that are activated by particular tasks or behaviors. And therefore that also fits in with our hypothesis that it's consistent with the bias in activity within the motor hierarchy for control of proximal and distal upper limb movement. So we're kind of therefore suggesting that neural circuits activated more so by each of the respective tasks and their associated behavioral demands are more likely to be facilitated by TDCS as they are more active at the time of the stimulation. And what we kind of hope is that these kind of results highlight potential new ways that stimulation can be used in maybe ameliorating motor declines in aging, but then also presenting some ideas about how we can potentially get more robust upper limb rehabilitation post-stroke, where we can target more so distal or proximal deficits. And then we'd just like to say a big thank you to supervisors, Ned Jenkinson, John Stuart Britton, Chris Marl, and then also co-author David Punt, and Ali Hall, lab member as well, and then all my funding and affiliation. And then just to plug finally, some of my supervisors have a funded opportunity here in the UK to do some kind of cool stuff with non-invasive brain stimulation and motor behavior. So if people are interested, they should definitely email Ned. Thank you.
Thank you, Matt. Does anyone have any questions off the bat that they'd like to jump in and ask?
I'm going to jump in. Yeah, I have a few. Thank you, first of all, for that really great talk. And I like both of these talks for just how rigorous they were, in dissecting these mechanisms. I actually did my postdoc in Birmingham. So I lived in the Northfield Council Flats for a while, if you know what that is. So I wanted to ask you, well, I guess two questions. Can you talk more about the timing? I mean, I know you turned on the TDCS even during the baseline and left it on. And does it matter? Do you think it might, is there some art there, or, either based on what you did or if you had, if you could run a million more subjects? And also, I mean, your final slide, you put mechanisms kind of question mark, but if you can say more about that, I mean, is there just like a naive mechanism just to say, well, it boosts and it interrupts and we don't need to think more about it? Or is there more there that you're interested in?
So with regards to timing, we were kind of tried to be constrained by those kind of initial previous studies that we'd shown reports on, trying to keep as closely to their protocols as possible. So yeah, it was started at the beginning of baseline. It ran through those and ran 17 minutes into the task, which ended at the adaptation phase. So it was a relatively non-specific protocol in terms of the timing of stimulation. And like I said, we tried to replicate some of the study designs as those previous papers, so we could not kind of add extra confounds. What we've done kind of since that work is look at how we can potentially more optimally apply stimulation in terms of, obviously these long kind of bouts of stimulation don't always seem to be specific to the behaviors, and during these tasks people are doing lots of different things, motor planning and kind of return movement. So we have done some work where we've shown that if you kind of apply stimulation in really short bouts, specifically kind of yoked to the specific movement you're looking to modulate, that there are some kind of nice effects there in terms of how we can kind of hone in on some of these stimulation protocols in terms of the temporal specificity. And then in terms of the mechanism, obviously in terms of how it's driving performance, we can either suggest that the stimulation by kind of enhancing the excitability and the firing rate of the neurons, whether you're increasing the performance of those movements and therefore that's driving better adaptation, or whether you're influencing the plasticity in the two different regions. And by doing that, you are influencing those kind of adaptive processes. It's quite difficult to separate with what we've got at the moment, but that's definitely something that we'd like to look at further in terms of how it's actually affecting the behavior rather than just kind of exciting, inhibiting. I think that's something we would like to kind of divulge further.
Any other questions for Matt?
If there isn't, I just want to jump in again. You mentioned the short bouts. So, or are you thinking maybe like you apply stimulation even for a very short period during, you don't need the many minutes. You just need to do it when it counts?
Yeah, so that's kind of fed off work from kind of Lynn Byman and that work where they showed that really short periods could influence kind of firing rate and spiking rate of neurons. So we designed a pretty complex context dependent adaptation task where, yeah, we stimulated for about 3-second epochs, which were specifically during, it was like a 1 second ramp, or 1 second hold, and a 1 second decrease, which I didn't have time to get into too much here, but it was specifically applied during kind of a 1 second movement, which was able to kind of enhance certain aspects of adaptation where we had a control movement which had no stimulation. And it showed that there was an enhancement when we applied these really temporal associated bouts of TDCS. So that's kind of watch this space kind of work.
That's really exciting. Yeah, I'll look forward to seeing that.
All right, we just have about a minute left. I did want to just say that Sarah put an answer to a question from Mark George in the chat. Dr. George, do you have any follow-up questions about that?
No, that sounds great. These were wonderful talks. This is a great journal club.
All right. If there are no other questions, we're just about at time, so we can wrap it up here. I want to give a big thank you to both of our speakers. You, both had wonderful talks, really interesting and exciting work. So thanks so much for taking the time to share that with us today. Thanks to everyone that attended. There will also, has been a recording of this, and we'll be sharing it. So if you have, colleagues that wanted to attend but couldn't, feel free to let them know about that. And then watch our Twitter account for when the next journal club will occur. Thanks, everyone. Hope you all have a great day.